Welcome to this video on how to get an A star in A-level biology series. This video is for CIE International A-level biology. So today's topic is 1.2, that is about cells, and in 1.2, before covering the part A, we are going to cover the B part in the curriculum, in the syllabus. And in the syllabus, B part is for the recognition of different eukaryotic cell structures, and then we need to know their functions. So different eukaryotic structures you can see on the right side of the screen. These are all 13 structures which you need to know according to the curriculum, and we are going to discuss these structures one by one. First, their presence in the cell, and then what their functions are. You only need to know, and you only have to focus on these 13 structures, because all the questions in paper 1, 2, or maybe in 3, they will be just focusing on these 13 structures. You probably see some other structures in your textbook, but there's no need to focus on any other structure because they are out of the syllabus. I'm going to minimize the right side window, so some features up till lysosomes will be visible at one point in time, and then we're going to discuss one by one about all of them. So as we know that the outermost covering of a cell, that is called as the cell membrane. In IGCSE we have learned that this is the cell membrane, but in AS level or in A levels we should not be calling it cell membrane, we should be calling it cell surface membrane. In some books you can find it is written as plasma membrane, but Cambridge University that prefer you to call it cell surface membrane. The examiners like Richard Fosbury, they prefer the term cell surface membrane. So the cell surface membrane, it could be seen, it's the outermost covering, and this cell surface membrane is the membrane which is made up of phospholipid bilayer. So this is a phospholipid bilayer. What does phospholipid bilayer mean? In more detail, we're going to cover that in a video for topic 4. The whole topic is about phospholipid bilayers. This phospholipid bilayer, its function is to regulate substances entering or exiting the cell. We're going to cover all of the organelles quickly, and then we're going to go and we're going to talk about their functions. So we have seen that the cell surface membrane, next to that is the nucleus, which is surrounded by nuclear envelope. So we can see that this cell is having a nucleus in the center, and this nucleus is also made up of cell surface membrane as its outer covering. The outer covering should be called as the nuclear envelope. So the nuclear envelope is the one that is enclosing the structures present inside, called as chromosomes. And chromosomes, generally they're not visible like that. They are X-shaped structures, but this shape cannot be seen at this point in time. I'm just drawing these structures so you can just remember it, that inside we have the DNA, but the DNA actually does not look like this. The chromosomes do not appear like this. At this stage of the life of cell cycle, because this cell is not undergoing any cell division, when the cell is not undergoing any cell division, the chromosomes are not visible. Then the chromosomes should be seen under the microscope during the cell division process only. Right now, the chromosomes are visible as just jumbled up threads over here, and a term is used to describe these chromosomes, which is called as chromatin. So this is chromatin, and chromatin refers to the total DNA content of the cell. This is chromatin, which is the total DNA content of the cell. The next structure which we're going to talk about, that is the rough endoplasmic reticulum. Endoplasmic reticula, they are very important structures within the cell, and we have two types of these endoplasmic reticula. How are endoplasmic reticula formed? Remember this thing, endoplasmic reticula are formed due to the further inclusions of the cell surface membrane. The new cell surface membrane keeps on adding, and then the old cell surface membrane, it keeps on transforming into endoplasmic reticula. So you can see it from this arrow that the new endoplasmic reticula, they keep on adding. We have two different types of endoplasmic reticula, and these two different types of endoplasmic reticula, they are rough endoplasmic reticula and smooth endoplasmic reticula. Both of these, they have slightly different function, which I'm going to tell you in a minute. The rough endoplasmic reticula are also referred as RER, and these are called rough endoplasmic reticula because their outer surface appears rough due to the presence of small tiny structures on them. These small tiny structures are actually the smallest organelles present in the cell. And these small tiny structures which are studded on the endoplasmic reticula, they are called as ribosomes. Ribosomes are generally involved in the synthesis of proteins, and these ribosomes could be of different sizes in different locations of a eukaryotic cell. The one which are attached on the rough endoplasmic reticula, these ribosomes are of size ATS. ATS is actually, S is a special unit which is used to describe very small sizes. This is a Svedberg unit. You don't need to memorize that. You can easily forget that. But S stands for Svedberg unit, sometimes wrongly read as 805. I have even seen some teachers calling it 805 and 705. It is not 705, this is 70S. S is the Svedberg unit. The ribosomes which are attached or which are studded on the rough endoplasmic reticula, which are making the endoplasmic reticula rough on the outer surface, these are involved in the synthesis of proteins which are supposed to be exported out of the cell. I'm going to cover that more in the details once we're going to talk about the two types of rough endoplasmic reticula in detail. And the ATS ribosomes which are attached on the rough endoplasmic reticula, they are involved in protein synthesis, and these proteins are usually exported out of the cell. And they are involved in many processes like the extracellular digestion, or they could be some other protein, they could be some functional proteins, they could be some structural proteins, but most of them, they have to pass through the Golgi apparatus before they are going to be exported out. The type of ribosomes which are attached on the RER, they are called as fixed ribosomes. These ATS fixed ribosomes, they synthesize proteins and then those proteins are exported out. We have some other types of ribosomes as well, and it can be easily understood that these ribosomes are actually not attached to the rough endoplasmic reticulum. These ribosomes are called as free ribosomes, and then these free ribosomes, they are involved in the synthesis of proteins which are to be used within the cell. Let's talk about another type of endoplasmic reticulum, which is called as smooth endoplasmic reticulum. As the name suggests, these endoplasmic reticula, these are not having any ribosomes studded or attached on their outer surface. That's why their outer surface, it appears smooth. These are involved in the synthesis, storage, and transport of lipids and steroid. Remember this thing, these are synthesis, storage, and transport of lipids and steroids. The steroids are usually the hormones, which are steroid hormones. For example, estrogen, progesterone, as the name even suggests that estrogen is the steroid hormone, progesterone is a steroid hormone, it's a steroid hormone. So generally, the smooth endoplasmic reticula, they are involved, they are abbreviated as SER, and they are involved in the SST of steroids and lipids. While RER, they are involved in the SST. SST stands for synthesis, storage, and transport of proteins. The next organelle which we're going to talk about is Golgi apparatus or Golgi bodies. These Golgi bodies, they are present very close to the rough endoplasmic reticula, and these Golgi bodies are involved in the packaging of proteins. All of those proteins which are made by fixed ribosomes attached on the rough endoplasmic reticula, these are going to be modified by the Golgi apparatus. Golgi apparatus is a stack of multiple membranes. We're going to talk about the details of the Golgi apparatus and how do they perform their function. They are usually enclosing the proteins in small vesicles. Next organelle which we're going to talk about is the nucleolus, and this nucleolus is playing very important role in the formation of ribosomes. Nucleolus could be one small spherical big structure found within the nucleus. Sometimes they could be two as well. If there are two, they are called as nucleoli. So nucleolus or nucleoli, they play a very important role in the formation of ribosomal RNA, and the ribosomal RNA then combines with a protein to make ribosomes. So the nucleoli are involved in the process of formation of ribosomes. Remember that these nucleoli disappear along with the nuclear envelope just in the beginning of the cell division. Now at this point in time, you can take a picture of the screen, and you can revise it through that picture. I'm gonna pause for 1 second so you can take a picture. The Mac users can take a screenshot directly from their computer, and if you're watching this video on your cell phone, you can take the picture right away. Next organelle which we're going to talk about is the mitochondria. Mitochondria are the sausage-shaped organelles, and these organelles, they are present in every cell, every plant and animal cell. Their main function is to generate ATPs, to form ATPs. Do not say produce energy. There's a common mistake students usually do. They say that mitochondria are involved in making energy. Energy can neither be created nor be destroyed. So never say that they make energy or they create energy. Yes, it could be said that they make ATPs or they release energy during the process of anaerobic respiration. A mitochondrion is also referred as a powerhouse of the cell. Remember that mitochondrion is singular and the plural is mitochondria. So we refer two or more as mitochondria and a singular is called as mitochondrion. Mitochondria are involved in aerobic respiration and they are the one in which the pyruvate, which is the end product of glycolysis, which is a stage of respiration, the pyruvate is broken down eventually into carbon dioxide and water and we release ATP. So synthesis of ATP or the release of energy is the function of mitochondria. Next organelle which we're going to talk about is the lysosome, and lysosome is involved in the process of storage of all the proteins which are made by the Golgi apparatus. The Golgi apparatus, they modify the proteins and then these proteins, they are enclosed in the lysosomes. Lysosomes are tiny vesicles and these vesicles, they are made by Golgi apparatus and they are usually containing hydrolytic enzymes. So after the process of packaging, the proteins are enclosed inside the lysosomes and then the lysosomes, they are responsible to sometimes transport these proteins to the cell surface membrane from where these are exported out during the process of exocytosis, or sometimes these lysosomes, they are just there in the cytoplasm and then they are separating the contents of the lysosome, which are hydrolytic enzymes, and these hydrolytic enzymes should not be coming in direct contact with the cytoplasmic contents, otherwise the cell contents in the cytoplasm are going to get digested, and this initiates a process. If this accidentally happens or this may happen according to a pre-programmed process called as apoptosis. We're going to talk about that later once we are going to go to study the lysosomes in much more depth. Let's talk about now centrioles and the microtubules. These centrioles and the associated microtubules, they also play a very important role in many functions in the cell, especially it acts like a center where the microtubules are organized. So centrioles are also called as microtubule organizing centers. A centriole is located very close to the nucleus, and it is not inside the nucleus. It is a plus-shaped organelle, and this whole organelle is made up of two cylindrical objects. I'm going to draw them separately outside the cell, and you can see that these are two cylindrical objects which are present at a right angle to each other. These two cylindrical structures as a whole, they're called as centrosome. Centrosome made up of two centrioles involved in two major important processes. Number one is the formation of spindle fibers during cell division, and the other process is the formation of cytoskeleton. Cytoskeleton is a structure present inside the cell only in the animal cell, because remember this thing that the centrosomes or the centrioles, they are only present in an animal cell. They are not present in the plant cell. Do plant cells not make spindle fibers? They do during the process of cell division, but the spindle fibers are formed by the cytoplasm in the plant cell. The cytoskeleton is a structure formed inside the animal cells to give structural support, a strength. We're going to talk about details of the cytoskeleton and the role of the centrosome as microtubule organizing center. Later, once we'll talk about the structure of different organelles and their functions in detail. The word cyto means cell. So the word cytoskeleton refers to the cell's skeleton. It's not a skeleton made up of bones or like the skeleton which we have. It is made up of microtubules, and this is giving extra strength or support to different organelles within the cell, because the animal cell does not have any cell wall to give any strengthening support, which the plant cells do have, as cell wall is made up of the hard dead material called as cellulose, which is a type of a polysaccharide. Now, before proceeding any further, I suggest you to take another screenshot at this point in time. Then I'm going to erase this animal cell, because the next organelles which we're going to talk about, all these organelles, they are present in the plant cell. They're not present in the animal cell except one organelle, which is the mitochondria. So the next organelles are chloroplast, cell wall, plasmodesmata, large permanent cell vacuole, and the tonoplasts. They are all associated with the plant cells. So take a screenshot right now, and then I'm going to erase this board, and we're going to look into the structures which are present in the plant cell. Now, about the plant cells, one thing is very clear, that plant cells also contain a cell surface membrane. Many students think that the plant cells only contain a cell wall and there is no cell surface membrane, which is not true. And also remember this thing, whenever you're drawing a cell wall, especially for paper 3, where you're supposed to draw a cell wall, it should be drawn in a double line, and whenever you're supposed to label it, start the labeling exactly in the center. Okay, so labeling should start from the middle of the two lines between the outer edge and the inner edge. Actually, the whole thing, the cell wall, it means that between the two lines, it is the cell wall, and this cell wall is made up of cellulose. The main function of the cell wall is to provide support to the plant cell, and it prevents it from bursting. So prevention of the plant cell from bursting is the main function of the cell wall. Cell wall is made up of cellulose. And how can we label the cell surface membrane if we are drawing a plant cell? A cell surface membrane is the line which is sticking to the inner side. Basically, it's a membrane, it's a very thin membrane made up of phospholipid bilayer, and this is sticking to the cell wall from the inside of the cell. Plant cells contain a large permanent vacuole, and mostly it is covering the central part of a plant cell, and this large central vacuole that is permanent is having a membrane which is called as tonoplast. So tonoplast is the name of the membrane which encloses the vacuole. Vacuole of the plant cell contains a solution which is a 2% salt solution called as cell sap. So inside the vacuole we have a solution called as cell sap, and this cell sap is 98% water and the remaining 2% salts. Other than the cell sap, there is nothing, no any organelle and no any other structure. So in a photomicrograph, the largest area which you can see in the photomicrograph, that is basically representing cell sap, or it is the inside of the sap vacuole. It's also called as sap vacuole or large permanent vacuole. Other than the vacuole, plant cells definitely also have the nucleus, and they also contain mitochondria. Many students don't realize that there is a nucleus as well. There are mitochondria, there are Golgi apparatus as well, which we have done in detail. Many times students do a mistake. They don't realize that there are Golgi apparatus, there are endoplasmic reticula, there are mitochondria in the plant cell as well. About the mitochondria, one important thing that you have to understand, the chloroplasts are larger than mitochondria, and how to identify a chloroplast in electron micrograph. The chloroplasts appear to have these grana which are lengthwise, like the lines which you are seeing. These are parallel to the surface lines at the longer edge of the organelle. While in the mitochondria, the lines which could be seen within the mitochondria. So this is a way how to recognize that either it is a mitochondria or it is a chloroplast. So mitochondrion, they are having lines which are not parallel lengthwise, they are vertical lines over there, which clearly indicates that this is representing the mitochondrion. Another important feature that should be remembered, that both of these structures, chloroplast and mitochondria, they contain ribosomes, but these ribosomes, they are slightly different in their size as compared to the ribosomes present in the animal cells or in the cytoplasm of the cell. So cytoplasm of this cell, it contains ATS ribosomes, while the ribosomes present inside the mitochondria or inside the chloroplast, they are smaller, they are 70S ribosomes. We're gonna talk about these in more detail and the other differences in the mitochondria and chloroplasts once we will talk about the structure of these organelles in more detail. The last organelle in this quick review of the whole topic is the plasmodesmata. Plasmodesmata is the structure which is only present in the plant cells, and it is a structure which is present between two plant cells. In order to understand the plasmodesmata, I'm going to draw another plant cell along with this cell, and this is going to explain the role of plasmodesmata. As you can see over here, I'm drawing another plant cell, and this plant cell is basically sticking to the previously drawn plant cell. This is the cell wall, but there are certain regions of the cell wall which become very thin, and this is a cytoplasmic junction. So plasmodesmata is that area between the two plant cells where the cell wall becomes very thin, and the cytoplasm of the two cells, they get close to each other, and then they are involved in a very rapid and more efficient exchange of materials between the cells. We'll talk about plasmodesmata in more detail in the review of all of the organelles one by one. So this particular video is very good to introduce the topic to yourself, and we have covered almost everything in the topic 1, including the structure of cell. Of course, you have to watch the next video, which is about the organelles and the details, and this one is a quick recap. This could be used before a test as a quick recap and to recall all of the facts if you have studied this topic long ago. So just before the exam or just before a test, you can watch this video again. This will be pretty helpful to recall all of the concepts about the main organelles. And in the next video, you're gonna watch all of the details of the organelles. So thank you very much for watching the video, and please don't forget to subscribe the channel and press the bell icon if you have not pressed it yet, and if you have not subscribed, please subscribe, and hopefully this video will be very beneficial to all of the students. Thank you very much and stay tuned.